In this lesson, we'll address a very important question in the neurosciences. What is a memory? And we're going to tell a story, so far as scientists understand the story, about how a memory is made and then what can happen when we forget. We've learned that there are two major forms of memory in the human brain: a working memory system, sort of the short-term buffer for information. So what we are consciously experiencing, that would be the contents of our working memory system. And this is what we're thinking about or perceiving in the moment. And then we have the cerebral cortex, where scientists think lots of our long-term memories are stored. We also learned in a previous lesson that having lots of strong memories for information about our world makes us better thinkers. In addition, the more we learn, the easier it is to learn more. At the level of neurons, of course, brains are composed of neurons. We learned that there are different arrangements that neurons can take. They can be arranged into chains, like you see here, or we can have neurons that send their information to two separate areas of the brain. So two different areas of the brain can be processing that information. And then we have arrangements where two different parts of the brain, the neurons, can carry information to a third region of the brain. So we get convergence of information in this third region here. So lots of different complicated ways that neurons can be arranged, and the arrangements of neurons contributes to the function of those neurons, what they're doing, what kind of information processing they're doing. In fact, this third one down here is what we saw in the simple case of classical conditioning, where the brain is going to learn to associate a bell with the presentation of food. And in that case, we had the bell pathway and the food pathway converging on a target neuron here. And as a result of training, what we saw was the growth of this synapse right here in the bell pathway. So now after training, the dog will salivate to the sound of the bell alone. That growth there, that physical change, that biological change that occurred during the training period, this is what represents the learned information. We call that the memory trace. So there's the memory right there. So what is a memory? Well, it's this change that has happened, this biological growth at synapses as a result of the learning experience, and that change then comes to represent the learned information. What did the animal learn? The association between the bell and food. So now let's tell the story of sort of how a memory is made, but first we'll just give a provisional definition for what is a memory. We'll say a pattern of strengthened synapses among a set of neurons activated by the learning event and which represents the learned information. And if those strengthened synapses are maintained for a good period of time, then we'll be able to hopefully successfully reactivate that information. We'll retrieve a memory because those strengthened synapses have endured. So that's what it means to make a long-term memory. We're going to have long-term strengthening of synapses that represents the learned information so that we can retrieve that information later on. Now we're going to use this diagram to show the three stages in the formation of a memory. And the first stage is we'll call the encoding stage. And on the right, we're going to show what's going on with a memory at the level of neurons. And then on the left, we'll just show a patch of the cerebral cortex where some memory trace has been formed. So here again, the cerebral cortex would be the surface of the brain here. So we'll call that a cortical memory area. So during the encoding stage, a population of neurons is activated by the learning event. Over here on the right, we see the neurons that are activated are the orange ones with the little red marks. Notice not all of the neurons here are being activated. And so we get selective activity, and that's determined by the network wiring and the particular learning event that we are engaged in. For example, if this is in visual cortex, if I'm learning about giraffes, well, then there's going to be a certain pattern of activity in the visual cortex that would be different than if I were learning about sharks, right? So if we're seeing different things, we're going to have a different pattern of activity in that area of the brain. So we've got selective activity of neurons that are being activated by the learning event, and that's what we're going to call the encoding stage. The brain is responding to the learning event. Next, we have the all-important storage stage, where a subset of activated neurons grow stronger synapses, forming a memory trace. And we saw this in the case of classical conditioning. So up here, you'll notice the synapses are of sort of a weak state initially. They're small, and that's just sort of the baseline strength of synapses. But look at down here. As a result of the learning episode, we get biological growth of some of the synapses on their target cells. So we call that plasticity, the ability of neurons to change the strengths of their connections. And this change then is the memory trace. This change happened because of the learning experience. So learning involves the growth of synapses. Synapses get stronger, and the memory would be that pattern of synapses that got strengthened. This represents the learned information. And let's notice one more difference. The difference between how the word memory is written up here during the encoding stage and how it's written down here during the storage stage. You'll notice we have sort of a faint dashed letters here, and then more visible, more ink here. And that represents that the strength of the memory trace has increased as a result of the biological growth. So during encoding, the brain is making weak memory traces in the cortex, but over time, as this growth happens, the strength of that memory trace is increasing. And so it's written more darkly here. And then finally, in the retrieval stage, if we're attempting to retrieve this information, what we have then is an attempted reactivation of memory trace neurons, recreating the pattern of activity during learning. So over here now, we're putting the little explosion icons or whatever they are to indicate that when we're trying to retrieve a memory, we seek to reactivate the very same neurons that were activated originally during the learning event. And if we're successful at reactivating those neurons, then we can successfully retrieve the information. If we're unsuccessful in reactivating these neurons, then we will fail to retrieve that information, that memory. And I should mention here that as well as the increase in the strength of the memory trace as a result of this growth, every time we retrieve a memory, the brain actually gets to restore that information in a stronger way, so there could be continued growth of these synapses at the same locations. So we could really strengthen the memory trace even more as a result of repeated retrievals of the same information. And that, of course, has obvious implications in school, that one way to help build strong memories for information is to repeatedly retrieve that information. Now, let's tackle the question of why do we forget? There are several theoretical possibilities. Let's take them one at a time. First, here we've got some cortical patches here. These would be places where memory traces can be formed from a learning event. But let's say in this first example, why we might forget something is that we had a storage failure. We just didn't make a strong memory trace in the cortex to begin with. And over time then, when our brain seeks to reactivate that memory trace, we have to search for where that memory trace is. Our brain literally has to search for the correct information. And because of the weak memory trace here, we fail. So even though we might have sort of located the memory trace, it's a weak memory trace, and we just failed to retrieve that memory trace. So this would be one example of forgetting. We never really learned it well in the first place. At the level of synapses, take a look on the left here. So here we have the original population of neurons being activated by the learning event, and we have the growth of certain synapses. That's the memory trace. So that's storage success. But what if during the learning event we didn't make those important synapse changes? So this will be a storage failure. We failed to really make a lasting change that represents the learned information. So if you don't make a strong memory trace to begin with, the brain will have a hard time reactivating this population of cells to produce successful retrieval. So here's our first example of forgetting. We didn't make a strong memory trace in the first place. Second example, theoretical possibility, is that you'll notice the word memory here in this case is written with more ink here. So we made a strong memory trace, so storage success, but maybe over time the memory degrades. And so it degrades to an extent that then when we are searching for that information, we fail to reactivate the memory trace and we fail to retrieve the memory. So this then would be the case where we make a good memory initially during the learning event, but then over time the memory degrades. Now, how could that be happening? Well, again, over on the left, here's the standard situation. We form a nice memory trace. But what if over time those strengthened synapses weaken again? And so that would have the effect of weakening the memory trace, making it so that it would be hard to reactivate those neurons and retrieve the information. So theoretical possibility number 2 is that we make a strong memory trace for some information we learned, but that over time memories are such things that they can degrade over time, resulting in weak memory traces that even if we can properly locate the memory trace, we fail to properly reactivate those neurons and we fail to retrieve the information. Take a look at option number 3. We make a strong memory trace, so we have storage success, but over time we lose the ability to find that strong memory trace. So our search systems have lost access to the memory. It's there, it's strongly stored, but we'll have failed retrieval because we did not properly find the memory trace. A computer analogy might help to understand this third option. Perhaps you've had a situation where your computer gets damaged somehow and it's telling you that it can't find any of the data stored on the hard drive. Well, you take it to the computer repair shop and they do some kind of data recovery procedure. So the information was on your hard drive, it's just that your computer was not able to access that information. That would be like option 3. We made strong memory traces, but over time our search systems lost access, couldn't find that memory trace again. At the level of neurons, what would that look like? Well, on the left here's a situation where we're successfully reactivating the neurons involved in the memory trace. So this would be retrieval success. So the green arrows would be activity from other parts of the brain. The search systems, they successfully reactivated just those neurons that were involved in the memory trace so we can retrieve that information. But over here on the right, you see that the memory search is trying to activate other neurons that were not involved in the memory trace. And so we're going to fail to retrieve the desired information. Notice that the synapses are strong here, indicating a strong memory trace. It's just that our brain memory search systems can't find this population of neurons that represents the information that we want to retrieve. So we can give names to these 3 theoretical possible explanations for forgetting. We have the storage failure situation. We've got the idea that we can make strong memories, but then those memories, the very same memories, degrade over time. And then we can make strong memories, but our brain can lose access to those strong memories. So these would be 3 explanations for forgetting. Now, are they all happening in the brain? Well, scientists have good reason to eliminate option 2. They don't think option 2 is going on because that would undermine the very point of having a long-term memory system. If memories can simply just degrade over time, then we don't really have a long-term memory system. Let's look at it from the point of view of neurons again. On the left, we have the standard situation. We learn something and we make a memory trace. That involves making strong synapses among a set of neurons here. So that's the memory trace right there. Now, option 2 for forgetting says that these strong synapses, just over time, they weaken again. Well, then we don't really have a long-term memory situation, right? We want a system where once, as over here on the right, where once you make strong memory traces, we want to keep those synapses strong. That's what makes it a long-term memory system. So just from a theoretical point of view, scientists don't think option 2 makes sense for understanding forgetting because it then wouldn't make sense that we have a long-term memory system. So that leaves options 1 and 3. So from the current point of view, scientists think that forgetting can be about 2 processes. 1, we never really made a strong memory trace to begin with, and then we failed to retrieve that memory trace. 2, we did make a strong memory trace, but we have not used or retrieved that information in a long period of time, and our brain has lost access to that strong memory trace. So in conclusion then, what is a memory? Well, we've broken it down into 3 stages: the encoding stage, our brain is responding; the storage stage, there's biological growth; retrieval stage, we're trying to reactivate those neurons. And what is it to forget information? Well, we didn't make a strong memory trace in the first place, or we did and we just haven't used that information in a long time, so we've lost access to that information.